Okay. Respected chairperson, senior doctors, my doctor friends, I'm here to make a presentation on mitochondria as therapeutic target for diabetes with a particular focus on evidence-based findings. I thank Dr. Mohan and his team members for their kind invitations and also thank our chairperson for their introductions. My disclosure is what I'm going to do for another 20 minutes. The contents of my talk plan includes introduction, structure, function, role of mitochondria in diabetes, mitochondrial targeted therapeutic strategies for diabetes, and of course summary. So mitochondria, the powerhouse of the cell, as highlighted by Professor Dr. Krishna Ji. They are responsible for generating an ATP through oxidative phosphorylation. So mitochondrial dysfunction characterized by impaired energy production, reactive oxygen species production, altered mitochondrial dynamics has been implicated in the onset and progression of diabetes and its complications. So mutations in mitochondrial DNA and nuclear DNA can disrupt mitochondrial function, leading to the development of diabetes. Epigenetic modifications can influence mitochondrial gene expression, impacting mitochondrial function, contributing to diabetes pathogenesis. So, mitochondrial targeted therapies aim to preserve mitochondrial health, improve oxidative capacity, and ameliorate metabolic disturbance in diabetes. So, structure. So, the term mitochondrion derived from Greek word mitosis, which means thread, and chondrion, which means granule. So, mitochondria are rod-shaped, double membrane bound organelles, size ranging from 0.5 to 1 micrometer in diameter, and if you look into the structure, it has an outer membrane, inner membrane. The intermembrane separated by intermembranous space, and intermembrane is thrown to several folds to increase the surface area, which are concerned with electron transfer, oxidative phosphorylation, as well as nucleic acid and protein synthesis. So the structure of the crystal. So you can see number of folds, and we give the stalk and the spear. They are concerned with ATP synthesis. So oxidative phosphorylation. So mitochondrial dynamics involves mitochondrial fusion, fission, as well as mitophagy. So mitochondria are not static organelles. They are rather dynamic, constantly fusing with one another, mitochondrial fusion, and splitting into two, what we call it mitochondrial fission. So mitochondrial fission brings mitochondrial membrane constriction and fission. Next is mitophagy. That is a selective autophagy. It helps to maintain healthy mitochondrial population. In diabetes, impaired mitophagy contributes to accumulation of dysfunctional mitochondria, leading to oxidative stress and insulin resistance. So, before mitophagy, you can come across mitochondrial fission, so splitting into two, so several divisions of the mitochondria with varying sizes are formed. So, it is preceded by mitochondrial division, which generates individual mitochondrial fragments of feasible size. Next is mitochondrial biology. So mitochondrial dynamics, including fusion, fission, and mitophagy, plays a significant role in pathophysiology of diabetes. So dysregulation contributes to dysfunction, and that results, which is a hallmark of diabetes. So studies have shown enhancing mitochondrial fission, inhibiting mitochondrial fission, and improving mitophagy, that improves insulin sensitivity in diabetic models. So inhibiting excessive mitochondrial fission, enhanced mitophagy have been proposed as a therapeutic approach to improve mitochondrial function. So next is function. So mitochondria are involved in a number of key biological processes, starting from energy production, redox signaling, calcium homeostasis, mitophagy, immune responses, inflammation, and senescence. So mitochondria plays an important role in synthesis of ATP, also participate in energy production through fatty acid oxidation, play an important role in metabolism of amino acids, including the synthesis and degradation of fatty acids and cholesterol. So this particular slide is very important. So we have the transmembrane complexes, what we call it, the respiratory chain, that is electron transport chain 1, 2, 3, 4, and 5. So we have the generation of ATP. So electrons derived from oxidizable substrate, they pass through mitochondrial transmembrane complexes. The energy derived from the proton gradient leads to ATP synthesis and via chemiosmosis. If you look into the metabolic versatility, it contributes to energy production, which is essential for the formation of essential molecules such as hormones and also membranes, also plays an important role in calcium homeostasis, also plays an important role in apoptosis. So this particular slide was also shown by Professor Dr. Krishna G.Seshadri. So we have the TCA cycle that generates NADH and FADH, and that supplies electron, and electrons are transported from class 1, class 2, electron transport chain, and also class 3 as well as class 4. So during the process, some of the electrons are pumped into the intermembranous space. That creates a gradient, proton gradient. So type 5, that is the transmembrane complex, that plays an important role in ATP generation through the gradient generated by the electron which are being pumped into the intermembranous space. So under normal physiological condition, oxidative states are vital for various cellular functions, including signal transduction. Next is also maintains the cellular homeostasis. It also helps in the regulation process such as gene expression, cell proliferation, as well as apoptosis. However, an imbalance in the generation and detoxification of reactive oxygen species can lead to a state known as oxidative stress. So if you look into this particular slide, you can see during the process of electron transport, some of the protons are pumped into the intermembranous spaces. They are converted into oxygen, resulting in the reactive oxygen species production. And very important thing is, it also plays an important role in apoptosis also. So to counteract the harmful effects of the excessive ROAs, cells are equipped with a range of antioxidants. We get the enzymatic, non-enzymatic. Enzymatic includes superoxide dismutase, catalase, glutathione peroxidase, non-enzymatic substances like vitamin C, vitamin E. Antioxidants, they neutralize reactive oxygen species. So reactive oxygen species, it has a dual role, both as essential signaling molecules and as a potential source of cellular damage. That is crucial in the study of cellular physiology and pathophysiology. Next is apoptosis. Mitochondria can release cytochrome C, which activates caspases and triggers apoptosis. Next is role of mitochondria in diabetes. So basically, very important thing is there is impaired oxidative capacity. So that results in defective uptake of fatty acid, defective fatty acid oxidation, lipotoxicity, and especially with reference to muscles, it interferes with insulin signaling, causing insulin resistance. So mitochondrial dysfunction contributes to insulin resistance and impaired insulin secretion in patients with the diabetes. Next is excessive reactive oxygen species production, oxidative stress. Next is alteration with reference to mitochondrial dynamics, fission, fusion, as well as autophagy or mitophagy. They play an important role, that is impaired. They produce oxidative damage, various cellular components, leading to the activation of stress-sensitive pathways that impair insulin oxidation. Again, we have the antioxidants and we have the reactive oxygen species. They are balanced under normal physiological conditions. In pathological conditions, the balance is tilted towards more of excessive ROS production. So ROS are mainly generated at complexes 1 and 3 as byproducts of ATP generation. Under physiological conditions, ROS generation is balanced by compensatory enzymatic and non-enzymatic antioxidant defenses. So mitochondrial dysfunctions, its role with reference to beta cell dysfunction. So there is impaired energy production with an impact on insulin secretion. Next is impaired oxidative phosphorylation. Very important thing is oxidative stress. That can also result in the apoptosis of the beta cell. Next is alteration with reference to mitochondrial dynamics, including fission, fusion, and mitophagy. There is an imbalance between the fission and fusion process, which can lead to mitochondrial fragmentation and ultimately resulting in beta cell dysfunction. Next is speak of the mitokines. They act as signaling molecules. They regulate various metabolic processes, including glucose and lipid metabolism, which are crucial in the pathogenesis of diabetes. We have the fibroblast growth factor, which improves insulin sensitivity. GDF15, the levels are elevated in patients with the diabetes. So targeting these mitokines could lead to novel treatment for diabetes. Next is speak of the nuclear DNA and mitochondrial DNA. So mitochondrial DNA is highly prone to mutations due to limited repair mechanisms, very important. So it can impact oxidative phosphorylation, resulting in reduced ATP production and increased ROS production. Certain mutations, that is mitochondrial DNA, have been specifically associated with the diabetes with mutations involving A3243G. It leads to mitochondrial diabetes, often accompanied by deafness. This was pointed out by Professor Dr. Krishna G.Seshadri, that is maternal inherited diabetes and deafness. So mechanisms with reference to impaired insulin secretion in mitochondrial diabetes, defective ATP production, so defective insulin secretion, hyperglycemia. So next is genetic aspects. Both genetic as well as the epigenetic mechanisms, modifications contribute to mitochondrial dysfunction and the development of diabetes by encompassing both nuclear as well as mitochondrial DNA mutations. So, pathogenesis of DNA. So, we have the different organs: pancreas, liver, skeletal muscles, adipose tissue. So, mitochondrial dysfunction, and that results in impaired mitochondrial ATP generation, oxidative stress, inflammation, and apoptosis, in turn results in hyperglycemia. With reference to pancreas, insufficient insulin secretion, and the liver, failure to suppress hepatic glucose production as well as gluconeogenesis. Next is hyperglycemia induces mitochondrial dysfunction, ultimately leading to diabetic complications. So, hyperglycemia results in mitochondrial abnormality, dysfunction, apoptosis, and diabetes-related complication. So, abnormal mitochondrial dynamics induced by hyperglycemia may be the pathogenesis and common basis for the genesis of chronic complications of diabetes. Next is mitochondrial targeted therapeutic strategies for diabetes. So, mitochondrial targeted therapies, they represent a promising approach. These strategies focus on prescribing mitochondrial health. So, ROS produced by mitochondria contribute significantly to cellular oxidative stress, a key factor in the pathogenesis of diabetes. So, targeting mitochondria, a great broom to fight type 2 diabetes. We have the exercise diet, pharmacotherapy with reference to biguanides, thiazolidinediones, GLP-1 receptor analogs, SGLT2 inhibitors, and mitochondria targeted molecules, natural compounds, mitochondria targeted antioxidants, and inhibitors of PTP opening. So, therapeutic approaches that tackle mitochondrial dysfunction are of interest, and different approaches could range from preventing dysfunction by reducing RAS production to modifying mitochondrial quality control mechanism. These are all the primary objectives. So, approaches that target physical activity that stimulates insulin sensitivity by improving the mitochondrial function, very important. So, mitochondrial turnover, mitochondrial biogenesis, improvement with reference to the respiration. So, next is physical activity, also activates PGC1 alpha enzyme. This upregulation is actually, it plays an important role in new mitochondria within cells. The enhanced mitochondrial biogenesis has been associated with numerous metabolic benefits. By increasing the number of mitochondria, exercise improves the capacity for oxidative proliferation. Resveratrol has been found to enhance mitochondrial biogenesis, and ICOR has shown promising results in reducing peripheral insulin resistance. So, regular exercise acts as a potent physiological regulator of mitochondrial biogenesis. So, mitochondrial targeted therapies. So, agents that target, that is reactive oxygen species, stimulate mitochondrial biogenesis, modulate mitochondrial dynamics. So, antioxidants targeting mitochondria can attenuate oxidative stress. Enhance mitochondrial biogenesis can improve mitochondrial function. Mitochondrial dynamics influence mitochondrial as well as cell health. So, these are all the antioxidant molecules that have been tried in animal experiments as well as human only for a shorter period of time. So, these MitoQ, SKQ1 and SS peptides are examples of mitochondrial targeted antioxidants that have shown effectiveness in experimental models of diabetes. Next is mitophagy in patients with the type 2 diabetes. So, under normal physiological condition, dysfunctional mitochondria, they are eliminated by degradation, whereas in patients with the type 2 diabetes, they are accumulated. That results in apoptosis and also induces glucotoxicity and various other pathogenic mechanisms underlying pathophysiology of type 2 diabetes. So, compounds that modulate mitochondrial dynamics. So, mitophagy is a selective degradation. Urolithin A, natural metabolite derived from ellagic acid, which are nothing but, that is acid obtained from fruits or vegetables, has been shown to enhance mitophagy. Pharmacological interventions on mitochondrial dynamics. Bigger mitochondrial dynamics modulators, antidiabetic medications like sodium glucose SGLT2 inhibitors or other antioxidants. So, according to the perturbation of mitochondrial dynamics and function, pharmacological interventions had an impact on those processes, could be categorized into three main groups, including dynamics modulators, antidiabetic medications, and so on. So, improving the progression of diabetes and its complications by targeting mitochondrial dynamics with reference to diabetic nephropathy, cardiomyopathy, diabetic retinopathy, as well as peripheral neuropathy. So, what are the clinical applicability? So, we have antioxidants currently available. So, what are the limitations, potential challenges? Lack of large clinical trials. So, ability to confirm the efficacy and safety. Antioxidants can mitigate oxidative stress and improve mitochondrial, but excessive antioxidant supplementation may lead to
Classically,
reduced
only insulin
stress
is,
and potential cellular damage. And it is important to determine the optimal doses as well as the treatment duration. I told you it has a double membrane structure. So the delivery into the mitochondria, that is a challenge. That is a question mark. So unique dual mechanism structure, innovative approaches to ensure successful targeting, and strategies such as nanoparticle-based delivery system, lipophilic cations or peptide-based carriers are being explored. And there are intra-individual variability in treatment response can limit the effectiveness of the mitochondrial targeted therapies for all patients. Effect of metformin on mitochondrial function. So we have the mitochondrium, it induces mild inhibition of complex 1, very important. That results in increased AMP. That inhibits the enzymes involved in gluconeogenesis. It also inhibits the mitochondrial, that is glucose 6-3-phosphate dehydrogenase, glycerol 3-phosphate dehydrogenase, and suppresses gluconeogenesis, and also causes stimulation of the AMPK as well as increases the levels of the GLP-1. Metformin raises hepatic redox state. Metformin inhibits complex 4, which can result in an indirect inhibition of this particular glycerol 3-phosphate dehydrogenase that suppresses gluconeogenesis. That leads to the inactivity of NADH. So it fails to convert lactate into pyruvate. So effect of metformin on mitochondrial function in mitophagy. So in short, it improves mitochondrial function. It reduces oxidative stress, and it improves the oxidative phosphorylation. So glitazones and mitochondrial biogenesis. Glitazone it plays an important role in modulating the various functions of the mitochondria. SGLT2 inhibitor, it reduces the mitochondrial ROS production by 3 ways. Inhibit effects on hyperglycemia, NADH level, and reduction in mitochondrial calcium overload. And switching fuel utilization from glucose to ketone bodies to free fatty acid to amino acids. There is upregulation of metabolic genes, increase in voltage gated sodium currents. So in patients with the diabetes, hyperglycemia. SGLT2 inhibitors improve mitochondrial dysfunction. So SGLT2 inhibition improves endothelial function and reduces mitochondrial oxidative stress. So overall, it improves mitochondrial biogenesis, bioenergetics status, and oxidative stress. Thereby, it improves mitochondrial functions. These are all the approaches to ameliorate abnormal mitochondrial function and to reduce oxidative production in diabetic kidney disease. Right. So, multifaceted actions of imiglamin. Imiglamin improves pancreatic function, glucose-stimulated insulin secretion, improves insulin sensitivity, and in turn improves the beta cell function. These are all the mechanisms of actions of imiglamin, unlike mitochondria, unlike metformin. So it partially inhibits complex 1, reduces reactive oxygen species formation, restores complex 3 function, prevents the PTP opening, thereby it prevents the pro-apoptotic cytokines. It improves beta cell function and enhances insulin action. And next is glucose lowering mechanism of imiglamin. Action on the liver, action on the adipose tissue, action on the skeletal muscles, as well as on the pancreas. So possible molecular mechanisms by which imiglamin improves glucose homeostasis. It improves insulin sensitivity, reduces oxidative stress, improves mitochondrial function. So effects of imiglamin on endothelial function, heart, kidneys, and brain. In short, mitochondria are widely considered as a power hub. Dysfunction as well as disruption of the cellular bioenergetics have been shown to be ubiquitous in type 2 diabetes. Understanding the molecular mechanism underlying the interplay between mitochondria diabetes, including the role of mitochondrial dynamics, biogenesis, and function is crucial. Bigonides and different agents, they improve mitochondrial function, and novel mitochondria targeted molecules, including natural compounds, mitochondria targeted antioxidants, positively impact on mitochondrial function and its quality control mechanisms. So before I conclude, let me invite and request all the delegates to register for this 53rd annual conference of RSSDA that is going to be conducted in Kochi from November 6 to 9. Please do attend. And yet another exciting event is the Tamil Nadu chapter RSSDA annual conference that is going to be conducted in Coimbatore on August 9th and 10th. I request every member to attend, make this a memorable and a successful event. Thank you very much for your kind listening.